have to say, you make me feel like really good. It was a wonderful day yesterday, and I hope everybody enjoyed it. I was so impressed with the diversity of the presentations, the creativity, just the heartfelt nature of every one of them, and I hope you felt it back. So this morning, I'm really pleased to introduce Bob Kuhn. I think it was 2 or 3 years ago, every time COVID has a time accordion, as Chris told me, stuff gets stretched out and smooshed down. But I get an email or a phone call from somebody from Georgia, a high school teacher from Georgia who wants to do phage discoveries. Like, all of you know how much it took for you to get to doing phage discovery and how much support and help. What is it, 10 years? 18 years? So I'm like, that's awesome. Sure. You can do this? And I guess I was right. So I introduced you to Bob Kuhn from Afar, Georgia.
I'm very happy to be here. I have to say before I get into this that yesterday was really heartwarming. I mean, not being from Montana, just the passion and the caring about your students and each other, it was really great. And coming to a conference, not really knowing who's going to be there and seeing the relationships you all have built was really great for me. So I'm really excited to go back to school tomorrow and take that energy with me. So, I call this Phage Friends because the way, and I'll talk about this a little more, but the way I do it right now is through a club. And I have 2 small groups of students that are looking for phages. And as you'll see kind of through this, I have some quotes from the students. Really, to them, the most important thing has been the relationships they've built in their team over this time. And it was not something I expected. At our school, I'm not sometimes sure who knows each other. We're not a community school. So that will be kind of a reoccurring theme throughout the presentation. Okay, and the other thing is that, so we've been doing this for 3 years now, and I'll talk more later about, I try to be very problem focused and phenomenon focused, like a lot of you all have mentioned you do as well. So just some history. I grew up in southwestern Pennsylvania, about probably 50 minutes from Pittsburgh. All of my people are from Pittsburgh. And this is where I grew up. This is the house. This is today's version of the house. It wasn't like this. You could literally go into the backyard, move some dirt out of the way, and pick coal out of the ground. There was so much coal. So probably 5 or 6 miles away were mines, underground mines. Pittsburgh and Butte, I feel have a lot in common in a lot of ways. So anyway, that's where I grew up. And then we moved, when I was 10, we moved to Northern Virginia, which was a very big shock for me, having lived in rural Pennsylvania. And I could have been one of your students in 7th grade. That was my first time I really got interested in science. My 7th grade life science teacher had a lot of classroom pets, and I could walk to the school like a lot of us could. And so you had your week if you wanted to sign up to come in and feed the pets in the morning. So snakes and turtles and all these things, guinea pigs. And so I came in one day and I noticed there were all these microscopes set up all over the lab benches and stuff. And I was curious about what was in the little pools of water that were in these little dishes. And unfortunately, I can't remember her name, but she was very good at what she did. She didn't tell me, she just told me to sit down and take a look. And the first thing I ever saw microscopically was Volvox. And when I saw that and then moved around and saw paramecia, euglena, different things. I was blown away that there was this microscopic world that I knew nothing about, that I was around all the time, but here they were. And it really kind of shook me up a little bit, like, because, I would swim in lakes and things like that, and these things are all around. Like, it really kind of nerved me for a little bit. But that got me kind of on my way to wanting to do some sort of science. I wasn't a great student in school. I was kind of a invisible kind of kid in high school. I relate to a lot of kids I see at my high school that remind me of myself. Didn't really have a big direction. And eventually that led me to Northern Virginia Community College. And I spent a year there getting my act together, and eventually transferred to the University of Georgia. I've been in Georgia ever since I went to college. And at the University of Georgia, I for what, I don't know why, I ended up being a paleontology major. And I probably should have switched to biology, but I did a research for undergraduates, kind of like mini thesis thing, and it was funded. So I felt like if I switched, I was going to be starting over. So there was money there, so I just went along with it. And that was in the Georgia salt marshes. So I figured this was the biological system, and I could work this to my advantage, so I didn't have to study rocks. I could study things that are preserved in rocks but have a modern equivalent. And that's kind of how I spun it. So I ended up doing a biology experiment for my undergraduate research, and then that transitioned into my master's research at the University of Georgia with my advisor, Sue Goldstein. And here we are, dirty from working in the marsh one day. And that was an incredible learning experience for me, because she and my other two advisors were very, not mean hard, but they were very specific editors and really wanted me to learn how to do science. At that time, I had no idea what I was going to do with this. But eventually that served me well when I became a teacher, because I knew how to, at that point, I knew what science was, and I knew how to do experiments and projects, and I could translate that over to teaching students. Didn't know that then. At that point, I was just trying to get my thesis published and reduce the amount of red on my papers every single time I got it back. After graduate school, I could not find a job in geology. I didn't really want one in geology. So I did what everybody did back in the 90s. We went into environmental science, and I worked for the Georgia Environmental Protection Division. Interestingly enough, back when I was in 7th grade, I took an aptitude test, a vocational test, and I didn't take it seriously. I just bubbled in randomly and stuff, because that's who I was back then. And it told me I was going to be a garbage man. And I didn't particularly like that, but I ended up working in the solid waste division of the state. And I had to get certified as a landfill operator. So I was a garbage man, so I fulfilled my destiny from 7th grade. That was an amazing job, but it didn't pay very much. So my wife, who was my girlfriend at the time, went to Korea to teach English, and just for a year to make some tax-free money for law school, which is what she wanted to do. And so when she came back, we decided that I didn't really like that job because it didn't pay. So when she went to law school at University of Georgia, I became a teacher because I enjoyed teaching when I was a graduate student, and I figured, hey, I'm going to get to do science every single day. I don't have to have the pressure of publishing, so I can pretty much do what I want. Research-wise with kids, I can teach other people, and that's how I ended up at Coyle Middle School. So I started out 6th grade, and then taught a year of 8th grade. We moved to Atlanta when she got a job, and I went to Centennial High School, where I was for 22 years. So I taught freshman biology and AP biology for about 20 years. And 3 years ago, our new STEM school opened, which I'll show you in a moment. and then I moved over to that school. Okay, so that's sort of like I don't know if that's a note card version of my history, but it's a little bit, since you don't know me very well. And so this is our school. This is Innovation Academy. It was just built about 5 years ago. It is one of 2 STEM magnet-focused schools. We don't have sports, we don't have arts. It is a career tech kind of pathway. We have an IT pathway, we have a healthcare pathway, and we have an engineering pathway. What we have in that school, I would say it's probably one of the most privileged schools I've ever seen in terms of access to things. They really did spend a lot of money on the school. We understand our privilege working there in terms of if you went to our South County equivalents, they don't have that there as much as we do. And certainly in our rural areas of Georgia, absolutely different. So we understand that this is kind of a unicorn kind of school. It does allow us to do a lot of things. Where it's located is just about 20 miles from Atlanta. One thing you have to understand about these northern suburbs is they're very wealthy and they're very competitive for students up there. So as a magnet school, we draw from about 62 different middle schools, and you don't have to pass any sort of tests or anything to get in. It's totally lottery. So we have a heterogeneous mix of kids. We're about 39% white, and then the rest non-white. So culturally, we're incredibly culturally diverse. We've had to rearrange our whole school calendar for so many holidays that we weren't even aware of for different folks' culture. It's been really great. But that's the outside of our school. This is looking from the second floor. So this is the biotech lab where I work right here. And this is where we eat lunch and have meetings. And this is the middle part of the school, and it's a big kind of rectangle around for 3 floors. So it's new and nice, and so this is the lab where I work. So this is the so, I know.
No pressure cookers in there.
Yeah, next conference is in this lab.
Yeah, I mean, like I said, I totally understand that this is not normal, and that, so there's 4 bench rows like that, and then we have some prep areas and stuff. These are a few of my biotech research kids. This is Audra, who is also my lab tech. She has a free period. Hopefully she's growing bacteria for me now, because it's about 3rd period and I need it for tomorrow. So she's more than capable. So this is the inside of the lab. It's a good place to work. So one of my students, Emily, who is a junior this year, this is the first quote where it's more geared towards her finding community in this process. So besides its amazing opportunity, this program, meaning Phages, has gifted me friends and meaningful relationships that I'll always be thankful for. So she's on a team of 4, which I'll show you in a moment. I call the older group Phage 1.0 and the younger group Phage 2.0 for no reason except for me to keep them straight. So we offer at our school a biotech pathway as part of healthcare. In the biotech pathway, we have 2 required classes, one called Biotech Essentials, and then one Biotech Application. So they're basically beginning and advanced biotech. And then we have a 3rd optional year, which is biotech research, where kids are doing independent projects. So this year I have 12 students, and that they're either working on teams or they're working independently or by themselves. So we have about 12 projects in that group. And then beyond this, how I actually got to the school was I was running at my other school something called DNA Club. So DNA Club is a molecular biology research club. And So I've had as many as 40 kids in that club, and we do more guided projects. So I'll have ideas, and I'll pull kids in on teams, and they work all year on these projects. These are some of my Biotech 1 kids from last year working on something in the lab. And then, there's, like I said, so diverse projects. Sometimes we have publishing options. Usually it's in the Journal of Emerging Investigators, the student peer-reviewed journal. So, and this is independently funded. So we have to get our own money, or we have to write grants. We did, for one of our projects I'll mention in a moment, we did a crowdfunding this year for sequencing. We got about $2,000 for sequencing to cover our costs. So we have to be really creative if we want to, because biotech stuff is expensive, as you know. So we have to be real creative in how we find this money. Okay, and this is the 6th year of the club. 3 years where I am now and 3 years at my old school. So the theme of this in biotech for me is all about the decrease in effectiveness of antibiotics. And so as a phenomenon, as a storyline kind of theme, in my biotech classes, I want longer projects that kids can demonstrate their biotech skills through the project. So and any of these, they have to do things like micropipetting, dilutions, PCR, gels, all these kinds of things. So I wanted projects that they could channel what they've learned through that, and I could assess them based on how they do in those projects. So in Biotech 1 this year, my sophomores have been doing Tiny Earth. It took me about 3 years to bust into this program because they were really hesitant of including people who weren't in college. And finally, last summer, I got to go to some professional development for it, and this is our first year doing it, and so far, so good. It's been a really fulfilling experience for them. This is from Phages DB, but I didn't have the Phages logo, but my goal for next year is to upscale the smaller, it's kind of like y'all do, bring Phages into my advanced class and have them do that as part of their curriculum next year. And then we already do it in our DNA club. And then for the future, to go along with that, so we've got antibiotic producing bacteria. We have phages that are infecting bacteria. And then for the PAIR project, they're about antibiotic resisting bacteria. So they all kind of go together, and the goal would be to wrap these things into these programs. So, all right, so real brief, DNA Club, these are kind of non-phage related projects. The non-phage related projects can get pretty complicated. So one of my other big passions is the bacteria Wolbachia. I don't know if you know much about it, but it's a reproductive parasite. It changes the reproduction of the host that it's in. So 70% of all arthropods are infected with Wolbachia. And we study it in one organism particularly, which is the common pill bug. We look at what are called sex ratio disorders in that pill bug. And so we look for a specific sex ratio disorder, which is pretty amazing when mom pill bugs from A. vulgare, Armadillididium vulgare species, the common pill bug, roly-poly, when she's infected with Wolbachia, all of her males that are male offspring, they can get feminized, they become females. So it actually changes them. So they're chromosomally male, but they develop as females. And so we've been tracking this. Our biggest project right now is looking at this across the United States. We have about 20 states last year that donated pill bugs from different schools and different people. And we've been doing this. This has never been done in the United States. It's only been done in Europe and Japan. And so we're looking at the frequency of these sex ratio disorders in the United States. We do not have any pill bugs from Montana. So if you would like to send me some, we can include you in our study. But we've done over 500 PCR reactions at this point, because for every single pill bug, you have to do 5 different primer sets to nail down this. So it's been a lot of work for the kids, and they can come in and do PCR on their own in the morning at this point. We have an apiary at our campus, just a couple hives. I would say it's one of the easiest things to generate phenomenon-based stuff if you know anybody who has expertise with bees or you want to learn. The main thing we do with this right now is we collect pollen that the bees bring in, which is what you see here, and we do metagenomic sequencing on the pollen to figure out what plants the bees are going to. And we put a report out to the city of Alpharetta, and they've actually started adjusting their landscaping towards what the actual bees are using. So that's been really cool. Yeah, we've done a little bit of bacterial evolution. We do some eDNA, so we have a forest fragment. We don't have beautiful, amazing forests like you do around here. I'm in the metro, so we have fragments of forests, or what we call forest, green space, really. But one in particularly Big Trees Forest Preserve is kind of a unique place. Is protected, so it has some of the old growth trees in the middle surrounded by development. And there's a couple streams that run through that. And so we wanted to know, since amphibians are very sensitive to environmental change, we wanted to know what salamanders were in those streams. So we worked with a researcher from Kennesaw State University in Georgia, who's a salamander expert. And we took water samples from the stream and did metabarcoding for salamanders specifically. And then we went out and collected salamanders and did small tail snips, and the kids do Sanger sequencing to identify, we could visually identify them, but to confirm the identification of the salamanders. So anyway, the point is we have a bunch of diverse projects we do in DNA Club, and that's been really fun. The Phage projects are part of that. So my first Phage team has been doing this for 3 years, and Phage 2, which was this year, my sophomores, they've just started this year, and they've been loving it. So, and you see a couple of my first group up here. So anyway, I'll show you what we're doing in Phages. It'll be pretty brief because I don't have the long history that a lot of you do, but we've had some success though, which has been really fun. So first of all, how did I get interested in this? Marisa kind of alluded to this, but it came from a chapter from Carl Zimmer's book, Planet of Viruses, The Enemy of Our Enemy. I read that and just was amazed. I like some of you through like biology teaching, I knew about the early phage experiments for the discovery of DNA and things like that, but didn't really think about them as part of the what they call the holobiont, or the global microbiome of things that are out there. And so that led me eventually to the Perfect Predator book, which I read just for fun. And then I thought about whether I could do this in my class. I reached out to C Phages, and to no fault of theirs, I think they told me no, but I think they told me no because they thought that I wanted to be part of their grant stuff. And maybe I didn't do a good job of communicating that I was just a high school teacher and I didn't want to be, I wasn't trying to infringe on anything. Anyway, so I don't take no for an answer real well. So, so I started just like Googling, and that's what led me to Clark Ford Watershed Education Program, just an info email. And I emailed that info and just said who I was. And then I got put in contact with Marisa, and and then the response was, well, this is not geographically limited. Like, you could do it where you are. And so we just started doing it, and
It'll be fine.
yeah. But I'll tell you that, like I said, this is the first time I've met, I think, all of you, but I've been using your media prep protocols for 3 years, not knowing who you were, but they've been invaluable to us. So thank you for that. That's been great. And of course, Phages DB has been a big help. And then, I've sent like thousands of emails to Marisa because I don't have a mentor. She's my mentor, right? So, all All right, so this is my first group. Megan, Emily, Needy, and Timmy. Megan is our senior. She'll be leaving us. She is special. She got a full ride to Mercer University, and she got some sort of special like governor's scholarship where they're going to give her 15 grand to do research at Mercer. I don't know what that means. So maybe she wants to be a doctor. So I don't know that it'll be phage research, but she knows how to do this stuff anyway. The other three will be back next year to do more. This was their first phage that they ever discovered. And so we called it Starbird because our mascot is a phoenix. So we're looking, and we have like a space theme to our school. So they decided to name it Starbird. And so this is one of the, their plaque plates they did for that. And then they eventually went on the second year to discover 4 more phages, one for each of them, which was nice. And one thing I stumbled on, because I was at the University of Georgia, I reached out to their electron microscope center, and I just inquired, like, how much would it be for us to get these photographed under the TEM. And Mary Brown Ard, who's been there for forever, for 25 years, she said, it'll cost you nothing, we just write it off. So we need educational outreach stuff. You need this, it's a nice quid pro quo, just come on in, we'll get the kids in here. And so during the second year, they actually taught them how to work the machine, and they let them drive the machine, which is pretty cool. And then next year, they said, don't send us your lysate, come in and you'll prep it yourself, and then you'll be able to do it right then. So that's been a pretty cool development. So this was their second year, and these were the phages they found their second year. And these are their lysates up here. And we've sent those in to Marisa, so those are in Phages DB. And so this is Megan's, kind of long, but as high school students, we're used to the idea of textbook and review. However, through the Phage program, we work directly with biology, developing new biotechnology skills, learning to overcome obstacles in science, and achieving many successes. It is this early hands-on experience that allows us to grow and become better scientists in the future. So, and this was Starbird close-up, so. And these were the other 4. So we couldn't name this one Agamemnon, which Needy really wanted to, but since her name is so strange, she just added it on there and it was fine for the database. So so very, they were very quick to see the difference, even though I think these are all from the same family of phages, especially the tails were very different. So, So Emily said, I love being able to unwind and chat with whoever's in the lab about my day or things we're excited about. I've become close to my team, and the Phage team has brought me closer to my school community. I'm extremely thankful that I got to spend the Phage program with these really amazing people. So our school can be kind of high pressure, and what they've found is they're doing this on their own after school. It's not part of school. So when they come in the lab after school, it's decompression for them. And that was an unexpected development. So they're very relaxed, they have a good time, and they've had some success, maybe because it's not such a pressure thing for them. This is my second team. These are my youngsters. So Sasha, Anish, and Bridget. And they started this year. They did not have the easy success that the other group did. We too had some contamination issues, and we had some top auger issues too for a while, but they figured it out. And this was, they found one phage this year, and their phage is actually the highest titer we've ever had. So it's a pretty amazing phage. So anyway, this is their phage. They named it Twig for whatever reason that kids name things, but yeah, we just imaged this a couple weeks ago, so this was a good one. So Sasha, who's a sophomore, she's driving the machine at UGA. It was mind-boggling to me that we had the chance to not only discover, but see our bacteriophage twig when no one else ever had before. I think that learning about how we can use our findings to help others fight infections has also expanded my idea of the influence of our discovery. Challenges real quick. We've had a lot of trouble getting enough phage titer to be able to extract DNA. The DNA extraction techniques, the one we use, which I think probably a lot of you use, we want to kind of learn some other techniques. Some of the other ones include chemicals that we can't really use. So I think we're going to try mag bead DNA extraction, try to boost titer with mag beads, maybe next year. We'll see. Sequencing costs for getting those sequenced are pretty high. And we're going to send 2 away because the juniors this year want to annotate their genomes next year as part of their project. So hopefully that can happen. And then the big thing for me that's a challenge is scaling up next year if I want to do this in my biotech classes. So I'll be going from 8 kids to 40 kids. And so just, media, equipment, that kind of stuff, that'll be a challenge. So Anish says, we were eventually able to overcome the challenge of contamination in our plates. This is done using various filters, regrowing the host bacteria to prevent contamination, proper aseptic technique, and doing many tests and controls to see what was causing contamination so we could prevent it. All right, and then the future, I mentioned sequencing the genomes of 2 of our phages, improving these methods, scaling up, as I mentioned. So feedback welcome, of course. If anybody wants to shoot me an email about how they do these things, I'd be more than grateful. And then lastly, Sasha said, not only have I loved learning about a completely new field of science, but I've enjoyed being able to develop some unique skills and explore different interests of mine. The community we've developed in the lab has been extremely heartwarming to me. Okay, so anyway, really my biggest thanks is to Marisa. I mean, I would not have been able to do any of this if she wasn't willing to spend time mentoring me through this. So it's been a pretty amazing journey for me. Here's my contact information if anybody wants to email, and I'm on Twitter. So or whatever they're calling it these days, if you're on there, look me up. I'll follow you back. The last thing down here is important to me from Bridget. It made me realize that you don't need to have a PhD or lots of experience to contribute to science. Everyone can contribute in small ways. There was talk yesterday about the young people here about becoming a teacher. I would say that if you're somebody who is just finishing up college and you're thinking about academia, but you're really not sure if you want to enter. I mean, it can be a lot of pressure to do things like publish and eventually tenure and things like that. Science teaching is a great way to scratch that itch without having those pressures. You can really do great projects, as you've seen with kids, and it is really fulfilling to be able to do that. So you take the best part of science research was experimentation and projects, and you get to do them, and it's okay if it doesn't come out all right. You don't have to worry about grant money or, your position or whatever. So it's definitely something to consider. And we have the same crisis that you all do, is that we don't have a lot of new teachers coming up through the pipeline. So it is definitely a consideration. I would just echo from yesterday. Anyway, hopefully I didn't go too long. Any questions that you have that I can answer? Yeah.
How many samples did your kids run to find those phages?
4.
4 samples?
But I'll tell you the trick, and Marisa alluded to it yesterday, the enrichment method, you will get phages almost every single time.
Except for me.
I mean, if you follow, you do the in-soil enrichment where you're growing